Should we go ahead and get started, or do we want to wait a few more minutes for folks to show up?
You can go ahead.
Okay.
We'll go ahead, Akshaya.
Oh, Lakshmi, can you hear me?
Yes, Ruchika.
Go ahead. You can
Okay,
start the
yeah.
introduction.
Okay, yeah. Okay, welcome you all for the evening session today. And I'm so happy and glad to introduce Beth to you all. Beth leads her image assay development team within the imaging platform in the Broad Institute of MIT and Harvard. Her team works with the biologists to help them create the image analysis workflow and makes the open source software Cel Profiler. And she joined Annie Carpenter's group in 2016 after completing a PhD in biochemistry and molecular biology at the University of California, San Francisco in Blackburn's lab, and she also hold a BA in biochemistry and molecular biology from Boston University. I'm very glad to invite you, Beth, for this session. Thank you very much. It's yours.
Yeah, I'm very excited to be here and honored to be invited, and really looking forward to talking to you guys. I'm going to tell you a little bit today about high content image analysis as done in Cel Profiler, which is a tool that's been in the imaging platform for about 15 years now. And then tell you a little bit more about why high content image analysis is so important to us, and what are the sorts of things that by extracting more features than you possibly ever even thought you could use actually can end up helping you do really novel and interesting kinds of experiments. So at this point, you guys have been in this course for several days now, so I probably don't have to convince you that images contain quite a lot of information and are really valuable data sources, just as much as, say, a sequencing experiment or any other kind of experiment. The fact that they can be quantitative and multiplexed is really valuable. But in terms of computational analysis of images, there's one aspect that I think a lot of times people overlook, which is just that even if it's something where you think you can count something by hand, human brains are in fact quite terrible at quantification. So when I first put this picture up, or if you have your window down relatively small in size, you might think that this is a full color picture. This is not. This is a picture that is mostly grayscale with a few colored lines drawn across it. And your brain, which is designed to give you the gestalt, the sort of overview of a situation really quickly, fills in the gaps because it's just trying to give you a sense of what is the whole picture. And so even in cases where you have a phenotype that you think is readily quantifiable just by looking at it, we really advocate for people to use these automated image analysis tools, because they provide more information, they provide quantifiable information, but also because your brain is not actually a very good quantification machine. And so that's why in our group we're very dedicated to building these easy to use, easy to get going with open source image analysis tools, because we generally never want someone to be counting things by hand. We always want people to be quantifying within software. And there are two major ways that in the imaging platform, which is made up of my lab, the Seminy lab, as well as the Carpenter Singh lab, run by Ann Carpenter and Shantanu Singh, that we think about image data and imaging experiments. The first is the one that you're probably most familiar with, which is the idea of measuring and scoring known phenotypes. I know what I want to find in my cells, and I'm going to do an experiment to look for it. And I'm going to talk about that first, and then I'll get into the doing morphological profiling and some of the sort of more finding your unknown experiments. So the tool that in our lab that we use for automated image analysis is a tool called Cel Profiler. Possibly some of you have worked with it, probably some of you have not. But it was started by Ann Carpenter and Ray Jones in 2004 and has continued to grow since that time. I mostly want to show this slide because I want to show the number of really excellent people who have worked on this software over the years. And we've almost always had a partnership between a software engineer and a biologist as we were writing it. And I think that's what helps make the tool very special, is it follows good software engineering practices, but also it's had heavy input from biologists over the years. And so we really try to frame things in the way a biologist would think, and make it so that it is easy to use as possible. The software is free and open source. You can run it on Windows, Mac, and Linux. And if you check out our website, you can download a copy. This is what the CellProfiler interface looks like for those of you who aren't familiar with it. And it's broken into two main parts. The left part is what we call the pipeline panel. And that really, I think, emphasizes some of the things that I think are most special about CellProfiler as a tool. What the pipeline panel shows is a list of operations that you're going to do on your imaging experiment. We call each one of these steps a module and the whole thing together a pipeline. So what this means is that we are creating essentially not just an image analysis, but a whole image analysis workflow. And that we can create these workflows just by dragging and dropping in our interface here different modules and rearrange them so that our workflow can be applied reproducibly to any image. You can also save this as a text file and send it to somebody else, or attach it as supplemental material on a manuscript. And again, the pipeline will say what version of CellProfiler made it. And so the way to actually sort of validate any analysis that you do is built right into the file. CellProfiler also defaults to whenever you save out the results of an analysis, it will save out a database table or a spreadsheet that says what is the pipeline that generated this data set. So reproducibility is really sort of baked in. And so you can use it on 1 or 2 images or several million images, whichever you happen to have. So as I said, on the left is the pipeline panel. And then on the right, we have the settings panel. And so it just gives you what are the configurable settings for whatever module you're looking at. Right now we're looking at the images module, and so it just shows a list of what are the images that are going to be analyzed in this experiment. But you could really, this can look quite different depending on which module. Some modules have only 1 or 2 configurable settings, some have dozens. If that sounds a little bit overwhelming, we agree that sometimes it is, which is why we have the #1 most important button in CellProfiler, which is the help button. Every individual module, as well as every individual setting with each individual module, has its own customized help. And so CellProfiler boasts about 70-something modules, and again, some of which have a couple settings and some of which have many. But we never want you to be lost as to what a particular thing is doing or why. And so you can always hit the question mark button, get some help, and it will tell you exactly what that setting does. For some of the ones that are a little bit more complicated, it gives you sort of recommended settings for your experiment type. We really try to make sure that even though there's a tremendous amount of information here, that you can still have a sense of what's going on. CellProfiler can be run in 2 modes. You can run in test mode, which is when you're sitting down at your computer, most of the face-to-face interaction you'll have with CellProfiler. This is where you can go through one image set at a time, where an image set is all of the channels that you took in one XYZ position. And so, you can have, 1 or dozens of channels, depending on your setup, and look to see how the modules that you've put in are running, how they affect what settings are coming out of CellProfiler. And so this is where you'll spend most of your face-to-face time. Once you're sure that your settings are good for your experiment, you can go ahead and hit the Analyze button, and then CellProfiler will automatically run multi-threaded on your machine. And again, for whether you have 1 image or hundreds or thousands or even millions of images. Our modules are broken across a number of different categories, and when you hit the Add Module button, you will see them in this category view. And this is just to make it a little bit easier to find what you care about. We have things like file processing, image processing, measurement, some things that are in a module we call advanced. Even what we have our worm toolbox, which has some special things specific for people who work on C. elegans. But since this even still is likely overwhelming a lot of the time, we've added a search function where you can search the modules for keywords. And so if I start typing size, because I know I want to find the size of some objects that I've identified, it'll show me that there are modules who have size in the name, like measure object size and shape. If nothing comes up with that in the name, say if I searched area instead, if I hit the search button, it will then pull up any module that has that word in the help functions. And so that makes it easy. Even if you don't know exactly what you want to do is called, you can search for it anyway. Typically, the first step of creating one of these CellProfiler workflows is to add some image processing modules. Again, likely you've gone over this already, but just to reemphasize, all classical image analysis segmentation works off of a single principle, essentially, which is my image should have the objects that I care about bright and everything else dark. And that's essentially the underlying principle for all classical image segmentation. And so for segmentation purposes only, any transformation you can think to do to your image is legal to get it to looking to this state so that you can find objects that you care about. Emphatically, this does not imply to images that you are going to be measuring, but images that are going to be used to do object detection or segmentation, where you're going to say, all right, this area of the image here is cell 1 and this area of the image here is cell 2. You can do any transformation you want in order to get this to happen. And there might be a number of things you need to do. You might need to mask out bright signal that is debris or dust that is brighter than the real things that you care about. Or you may need to enhance dim signal that really is there, but just is really close to the background level. And the more transformations that you do along the way to get to your segmentation, the more critical it is to eventually check your segmentations against your raw images. And we provide a couple different modules to do this in CellProfiler, one called Overlay Outlines, one called Overlay Objects, as well as something that we call the Workspace Viewer. And so Overlay Outlines and Overlay Objects behave basically the way you would expect. Here we have some nuclei that have been identified, and Overlay Outlines puts the outlines of them, and Overlay Objects puts object masks on top. But the Workspace Viewer I just want to highlight is a really nice tool. It was in CellProfiler 2, it was gone in CellProfiler 3, and now it's back in CellProfiler 4, where you can put together whatever channels that you want to look at, and however many channels you have, and then overlay the outlines of any objects that you found, and even measurements if you would like to put measurements on there. One of the nice things with the Workspace Viewer is, unlike the CellProfiler module windows, which will default to being all the way zoomed out every time you change a setting, the Workspace Viewer will stay docked and zoomed in on a particular area. So if you're iteratively trying to change a few settings to handle a tricky area of segmentation in one part of your image, the Workspace Viewer makes it really easy to do that. And CellProfiler has a number of these image processing modules that you can use to do these sorts of transformations to get to the point where you can find objects. I'm not going to read all of them. Many of them, again, are found in lots of other tools. I do want to highlight, though, the Run Image2 macro module, which if you have Image2 installed on your machine, which probably if you have CellProfiler, you almost certainly have Image2 installed. And if you're comfortable enough with Image2 to run a script or a macro to do something that you want to do that is in Image2 but isn't available in CellProfiler, you can take an Image2 script, point CellProfiler at your local copy of Image2, and you can incorporate Image2 steps into a CellProfiler pipeline. So we're really excited to have this functionality back in CellProfiler, and it can be really quite helpful if there's something that is a particular filter or transform that you just need to access without needing to write a whole CellProfiler module just to do it. Once you've found objects, you might also want to change the objects that you found in CellProfiler. And so CellProfiler offers object identification, which is typically done with the Identify Primary Objects module or the Watershed module. But you can also filter objects. So say I want to first find all of the cells and then filter them to only the cells that colocalize with a second separate channel, my GFP channel, for example, to pull out only GFP positive cells. You can edit the objects manually if your segmentation really is not working well and needs to be cleaned up manually. We generally don't recommend that you do this if you have hundreds or thousands of image sets, but it's possible to do. You can shrink objects down to their centers or expand them out by certain amounts of things. And you can even relate objects. So if I say find nuclei, and I also find nucleoli, and I want to count, say, how many nucleoli are in each nucleus, by using the relate objects module, I can create a relationship between those two where it tells me, for example, for each nucleus, how many nucleoli does it contain, and even if I want to, what is the mean of any measurements I've taken of the objects that are, it's what we call its child objects. So there are a lot of different things that you can do depending on how many kinds of objects that you care about in CellProfiler. But almost always your image analysis pipeline doesn't necessarily just end at finding objects. It then involves measuring them in some way. Sometimes a pipeline only needs counts, and that's absolutely fine. But very often you might want more measurements, and I'm going to talk later about exactly why you might want lots and lots and lots of measurements. And so in our measurement module, we have a lot of different things that you can measure on a whole image level or on individual object levels. I want to stress that many of these metrics can be found in lots of tools, not just CellProfiler. But the concept of just making it easy to create really big measurement suites, again, in a reproducible way, is, I think, one of the things that can be really valuable in CellProfiler. One class of measurements that lots and lots of pipelines contain are intensity metrics. And when you add the measure object intensity module, you don't just get, for example, the mean intensity, but you get the total intensity, the min and max, the mean, median, and standard deviation, and you can get object intensities from any channel. And so I can find nuclei in my DAPI channel or my hooks channel, measure their GFP intensity as long as, you know, those images were taken in the same location, and then do something like a filter to get only GFP positive nuclei, or just measure what is the GFP intensity in each of them. So you can mix and match channels and objects in any way that you want. Another very popular module to add for measurements is the measure object size and shape. And again, this doesn't just give you area, it gives you a lot of different things which are all mathematically defined in the module help, such as the perimeter, the eccentricity, essentially whether an object is more circular or more linear, the solidity, how many holes does it have, and even some things that are not as easy for the human brain to intuit, such as Zernike polynomials. So Zernike polynomials are high-level shape descriptors that essentially say, if I made this object a circle, how much does it look like the inside or the outside of the circle, or the left versus the right of the circle? And it's very difficult for a human brain to understand what a Zernike polynomial actually looks like. But if you just sum all of them up, they can give you really accurate descriptions of an object's shape. And so this is one of the first papers that showed that for biological images, Zernike polynomials could be really interesting. So here are a number of different cell shapes on the left, and then on the right is the description of the cell shape that you can get just by adding up some of the measured Zernike polynomials, and we actually measure even more than what's there. So you can see that we're not getting an exact measure of the cell size or the cell shape here. You know, the left and the right aren't exactly equivalent, but compared to simpler measures like the orientation and the perimeter, we're actually getting a much more detailed view of what each of these kinds of shapes look like. And so while these measurements are really difficult for human brains to intuit, they can be really valuable for higher level mathematical analyses of shape, which again, I'll talk more about in a little bit. Returning to things that are a little bit more human intuitable, we have a module where you can measure the intensity distribution of any individual channel within any individual object. So for example, if I divide the cell into concentric rings, how much is in each ring in the center going out to the edge? And likewise, how evenly distributed is my intensity within each ring? Is it pretty uniform all the way around? Or is it really different between, say, the left half and the right half? And so that allows you to get at things like, is my ER moving in a different location, or is my protein paranuclear, or is it somewhere totally else in the cell? And so we provide a lot of these measurements to allow you to do really fine-grained analyses. There are other ones I'm not going to talk about, such as measuring texture, measuring colocalization between different channels, and measuring things not just within objects, but at the whole image level. Eventually, once you've put together this nice Cel Profiler pipeline that finds the objects that you care about and measures them in some way that's interesting, again, whether that's count or whether that's something really sophisticated or both, we let you then save out images so you can create the outline and overlay images that I showed you earlier. And we, in fact, are really adamant that you should nearly always do this, because there's nothing more heartbreaking than thinking you found a cool quantitative result and then going back to the outline images and realizing that it is actually a result of poor segmentation. And so being able to quickly check after you run an analysis on dozens or hundreds or thousands or millions of images and check what the actual segmentation was can be really valuable and can save you from going down a road where you think you found something cool that was actually just a segmentation error. And you can export out your quantitative measurements with the export to spreadsheet and database modules to a bunch of different file formats. So if you're running on a cluster, you might want to save to a MySQL host that's running at your institution, or you can save out to something as simple as a CSV spreadsheet. And I've emphasized that this software can be used at a variety of scales, and we really just want you to do whatever works best for the experiment you happen to be doing. If you have a few to a few hundred images, you probably just want to run on your local copy of CellProfiler on your desktop or laptop. CellProfiler will automatically multi-thread, as I mentioned, so that it will automatically, if you have 8 CPUs, run 8 copies of CellProfiler at a time. So rather than analyzing one image at a time, you're doing 8. This number is configurable, so if you don't want to let CellProfiler take over all of your CPUs, you can give it a lower number, but it will automatically just try and make your analysis as fast as possible. If you have a few hundred or a few tens of thousands of images, you can talk to your local sysadmin about running on a cluster, and we have instructions on our blog and on our website about how to do this. And if you have tens of thousands or millions of images, you can consider cloud processing. And we, in fact, also support running on AWS directly with a package that we call Distributed Cel Profiler. So that's what I'm going to tell you about Cel Profiler. And I'm then going to shift into talking about, I made a big push just now on measurements and capturing lots of measurements. And a lot of times people might think, well, I don't need that many. I only need the 1 or 2 things that I know that I care about. But I would like to hopefully convince you over the sort of next half of this talk that, in fact, sometimes it can be valuable to capture measurements, even if you're not sure how they'll be useful. We call this morphological profiling. And it's really about finding the unknown unknowns in your biology and finding things that are not necessarily what you expected to look for. I want to emphasize, though, that in fact, the way most of biology is done is with the measuring of particular phenotypes. This paper is a few years old now, but I imagine it would probably be pretty similar if we did the same experiment today. Most papers, even papers that describe themselves as high content, only actually look at 1 or 2 measurements of the cells. And so a tremendous amount of really good biology is done just looking at 1 or 2 measurements at a time. And these numbers don't change too much when we look at a couple of other paper things as well. And again, if you can find the thing that you care about with 1 or 2 measurements, that's great. But in general, we've become a little obsessed with the idea that there's information that's actually hidden in images that, again, as I mentioned, human brains, which are not designed to be quantification machines after all, really can't access, but a computer might be able to, and might be able to give us some extra scientific information. So these are all bright field images of different cells in different cell cycle phases. Back in the time when I used to give this talk in person, I always took this point to ask, all right, who would like to tell me which cell is in which cell cycle phase? Never gotten anyone to raise their hand at that point. Because it's hard. I could probably have told you that these were anaphase and telophase, and based on that, I maybe could have guessed that these were in a sort of cell cycle sorted order. But it's definitely not obvious to our human brains that what these cell cycle phases are from purely bright field images. But a computer can actually tell this really accurately. So this experiment was done by the fact that these cells also had DAPI in them. They were sorted into cell cycle phases by DAPI by the human annotators, but then the computer was only allowed to measure them in the bright field channel. And looking at this confusion matrix where we're comparing what the actual cell cycle phase was and what the computer predicted, in general, the predictions are very, very good. And even the cases where we're seeing things off the diagonal, which indicate that the algorithm is likely confused, it's confused in a way that makes sense. For example, S phase, which comes between G1 and G2, those are the things it gets a little bit confused by. And of course, the transition from G1 into S and from S into G2 are not incredibly switch-like transitions. They are sort of gradual changes. And so the sorts of things mistakes that it makes are making sense. Likewise, prophase or G2 going into prophase is a transition that takes a while. And so the mistakes that it's making seem to be just as much a sort of artifact of the human necessity of dividing things into finite classes as opposed to just sort of showing a continuous process. And here's just a pretty spinning thing to show if we look at the feature space of these cells in a pretty spinny 3D thing, that things are clustering by their cell cycle phases in feature space too. And so this idea that there's information hiding in these images that our brains literally can't detect, but computers could, is what led the labs to create or want to really sort of dive into this field of what we call morphological profiling, which is the idea of just, if I, rather than measuring a cell in 1 or 2 ways, measure it in hundreds or thousands of ways, then I can create, again, almost like a microarray or an RNA-seq, whatever is popular these days in terms of looking at expressed transcripts. We can look at expressed phenotypes and looking at phenotypes of morphology and seeing which ones go up together and down together and how those correlate with biology. This slide implies, because often it's the case where this is done in stacks of multi-well plates with hundreds or thousands, but this approach doesn't need to be used at that scale. We've used this for cases where someone had 4 or 5 different mutants they were interested in looking at. You don't need, you can use this on a large scale screen, but you don't have to. You can use it on really whatever scale you're happening to work in. I said hundreds and thousands of measurements, and so you might ask, how on earth are you getting to that high of a number? Some of it is just that we break the cells down into their individual subcompartments. So we typically look at nuclei, cytoplasms, and then whole cells. And then we look at dozens of measures of shape, of size, dozens of measures of intensity, of textures. Again, all of the things that I showed you before, where CellProfiler will automatically create these large measurement suites. If you do that for enough compartments and enough channels, then you can get quite a large number of measurements. So our typical cell painting workflow these days, we're at about 5,500 measurements per cell. But does this actually work? So this was one of the first papers that led the lab to think that possibly it actually did. In this experiment, cells were treated with a number of drugs which had a known mechanism of action, and a number of mechanisms of action that looked really different. So protein synthesis and degradation versus DNA damage versus actin disruptors. And what we wanted to know was just, could a really simple imaging assay, in this case, a nuclear dye, an actin stain, and a tubulin stain, actually be able to take the drugs and classify them back into their mechanism of action classes just based on images alone. And what the lab found was that looking just at the nucleus, not even looking at the rest of the cell, but just the nucleus, you could actually get 67% accuracy of grouping these drugs into their drug classes with, again, looking here at the actual class versus the predicted class. Most of the time we're on the diagonal, indicating that the prediction is correct. If you go from just this one stain in the nucleus to all three stains, you get 94% accuracy. And so this, and again, similar to what I talked about before, the mistakes that it's making are kind of making sense. One of the sort of biggest confusions is between DNA damage inducing drugs and DNA replication altering drugs. But messing with DNA replication can cause DNA damage. And so these processes are super interlinked. And so it's maybe not that surprising that we're seeing confusion between them. Likewise, microtubule destabilizers, which can mess up mitosis, also can sometimes get confused with DNA damage, but messing with mitosis does cause DNA damage. And so the confusions, again, are more to do with the fact that we have a need to classify everything into a single class than that the algorithm doesn't know what it's doing. And so, again, being able to detect all of these different kinds of drug phenotypes with only sort of really simple stains, a DNA stain, an actin stain, and a tubulin stain, gave the lab some confidence that morphological profiling really was a valuable approach. The other thing that sort of really drove our lab into doing this was a paper that doesn't come from our lab, but led to a pharmaceutical company called Recursion, which my boss, Ann Carpenter, is on the board of, was a group that was at University of Utah looking at, they had a cell type with an siRNA that when you introduce the siRNA, caused a phenotype that was very similar to a disease state, specifically cerebral cavernous malformation, a stroke disorder. And the visual phenotype when you introduce that siRNA into cells is actually really quite striking. I think most of us would think that we could pretty readily distinguish between healthy cells that haven't been treated and these diseased cells that have been treated. They look visually quite striking. And so the experiment that they did was they added a whole bunch of drugs, and they tried to say which drugs, when added to knockdown cells, create the most normal phenotype. So I can screen to see if any drugs work well on this disease. And the really sort of exciting thing that they did is they both had a bunch of human experts go through the resulting photos and pick out the most healthy looking responses. And they had the computer go through and do a quantification of the morphological phenotype and have it pick out which drugs it thought created the most healthy phenotype. And then they gave both sets of drugs, the ones picked by human experts and the ones picked by the computer, to mice. And it turns out the mice do much better when they are given drugs that were picked by the computer. Because even when there is a striking visual phenotype to the human brain, it turns out it's not actually the one that's really helpful in determining whether or not the cells have truly reverted to a healthy state or not. Our eyes don't pick up on the right things. And so, again, this was really strong evidence of the idea that profiling could actually help identify drugs for diseases in a way that even a human expert could not. So this led the Carpenter Lab alongside the Stuart Schreiber's group to develop what we call the cell painting assay. We described this in a couple of different papers, and we're actually working on a next generation version of the protocol, which hopefully will be out in preprint in the next month or two. And the way this experiment was designed was just to be as inexpensive and accessible as possible and as easy to run on a standard imager as possible. So most of the dyes are things that you might already have in your freezer, things like Hooxt, Phalloidin, MitoTracker. And the idea was just, can we cram as many organelle-specific stains as possible into a 4-channel or 5-channel imager, so that we can just measure cells as richly as possible. So when I mentioned that we typically do 5,500 measurements, it's in this staining panel. The idea being that instead of looking for one piece of biology or one particular protein that we know we care about, let's just look at everything and see how the whole cell changes. Because again, we now have this idea that this hidden information might be valuable. And of course, I'm telling you this story, so hopefully you're going to be convinced that it works. So this was one of the next successes that the lab had, looking at overexpressed genes that we knew were involved in human health, and saying, if we do this cell painting assay, again, just looking for organelles, what can we actually classify in the same way we do drugs into mechanism of action? Can we classify genes into pathways? And so zooming in on this clustergram, here are a bunch of members of the RAF and RAS pathways. And in the next cluster over are some mutations, disease-causing mutations in BRAF and RAF. And you can see they're not clustering with the wild types, but they are clustering quite close to them. So this gave us some confidence that you could actually detect, not only could you sort genes correctly into pathways, but that the connections between individual pathways were actually informational. So Mohammed Roban, who was then a postdoc, now has his own lab in Sharif University in Iran, looked at the pathway-pathway interactions that he was able to find, and most of them were things that were already known in the literature, which again gave us confidence that this approach worked nicely, but is perhaps not the most exciting. But he did find one cluster, which was an anti-correlation between the NF-kappa B and Hippo pathways that was not known from the literature. And when we talked to experts in NF-kappa B biology or Hippo biology, they were like, nope, no idea that those should interact. And so we were able to convince them, since we're a totally computational lab, to build a reporter gene. And it turns out that when we look at a Hippo reporter, and then we overexpress NF-kappa B pathway members, that our reporter assay goes way, way down. And so we're actually able to detect a completely unknown pathway-pathway interaction purely from dyes like Hooxt and Phalloidin and MitoTracker. We're now working to extend this into disease states. And so this work is currently being led by Marzia Higigi, who's a great postdoc in the lab, looking at skin fibroblasts from patients with various psychiatric disorders. Again, I want to emphasize these are skin fibroblasts. These are not neurons. And what Marzia was able to show is when these are cell painted, and we look at the cell painting phenotypes for people with or without various psychiatric disorders, there are some differences that we can tell even just looking at their skin cells, which of course gives you tons of ideas about what you might do in terms of either screening tools for diagnosis or screening tools to figure out which drugs might actually help these people by looking for particular phenotypes that are or aren't there just from a very simple skin punch. And again, just to sort of drive home this, this is something we only really could have discovered with these sorts of morphological profiling approaches. This is the one feature, because this is work in progress, I can't actually tell you what it is, that really differentiates a couple of the different disorders. Depression from bipolar disorder in particular, this feature is quite different. But I've stared at these pictures for a very long time, and to my human eyes and human brain, there isn't a tremendous amount of difference that I can say is present between the cells on the left and the cells on the right. We can only see this particular pattern by looking at the really sort of fine-grained measurements. So in the last 10-ish minutes, I'm going to tell you a little bit more about how, now that we know that profiling works and can be used for some really cool ideas, how can we push it further? What else can we do with it? One thing that we're working to do is just to take this into 3D. So working with Nicholas Riveron's lab in the Netherlands, working to try to make 3D compatible versions of the cell painting assay. We're also looking at how incorporating information about the heterogeneity can improve profiling performance. So what I haven't gone into detail about is that in general, when we've been doing all of these profiling approaches, we're looking at the per well means, but per well means could express something very different. So the mean is exactly the same in both of these images here, but these actually have a very big difference in the variety of phenotypes that are present. And so Hamda Abbasi and Mohammed did some work on how we can incorporate information about the variability as well, and showed that in some cases we don't see a ton of extra ability to group things correctly based on adding that heterogeneity. But in certain data sets, and noting that this was a really large data set, we're actually able to group things into their known groups much more efficiently when heterogeneity was taken into account. One way that we're also hoping to push this further is to just make it easier to do and be more accurate in finding the cells and the nuclei themselves. And this comes from the fact that, again, I just spent 20 or so minutes at the beginning teaching you how to use CellProfiler and talking about how it's complicated and there's a lot of settings. And what if it wasn't? What if software just knew what a nucleus looked like? And so a couple of years ago, our lab helped create this data science bowl challenge where our lab hand annotated 28,000 nuclei. That took a while. That were at a number of different magnifications, and in some of them were fluorescent and some of them were histological, and across a number of different image types. And teams competed to create a universal nucleus detector to see, can we get away from doing things like having to have CellProfiler with lots of configurable settings. And compared to a reference created by a CellProfiler expert, most of the teams did worse, but some of the teams did better in a way that was not configurable. And if you gave it an image that looked like this, or like this, or like this, could just find the nuclei within it. And so this breaks down in the different classes, essentially how well it performed. Most of our images are what we call the small fluorescent class, because that's what's easy to get a lot of. But we had a data scientist and a cell profiler novice go through and do some analyses, and they did fine, but not amazing, with a lot of time. A cell profiler expert could do better faster, which makes sense, but the top performing model, without any need to do individual configuration, could do it better than any of those. And so NucleAIzer is a tool that was built off of that model, and the data that we created there is built into the training of some things you may have heard of, like Stardust and CellPose. And so these AI methods of finding nuclei in cells, I think, are going to become even more powerful. We're also looking to deep learning to not just find our objects, but also to find our features. So Con+Syot, who used to be a postdoc in the lab, who now has his own lab at the Broad, did a really cool experiment to try and help us use deep learning to find features, because features are, after all, just numbers. And I mentioned that some of them, like intensity, are really human intuitable, but some of them, like Zernike polynomials, are not. So we decided that if the features don't need to be human interpretable, we might as well use deep learning for them. So Juan came up with a really cool experiment where we're doing a training that's ultimately seems kind of stupid. We're training each cell to guess what drug it was treated with. And in that case, again, I say that's kind of stupid because we know what drug each cell was treated with. We treated the cells. But by training a network to try to guess for each cell which drug it was treated with, he trained a network that ultimately measured something interesting about what drug treatment does to cells. And so by then throwing out this ultimate end goal of guessing which drug a cell is treated with, because we always know that ahead of time, and just taking the network, he was able to treat the different parts of the network just like a texture feature or a shape feature or an intensity feature. And so that same experiment that I told you, we did 94% accurate with the 3 channels, the nuclei, actin, and tubulin. Juan's network was able to classify those cells 97% accurately and 700 times faster. And so we think that these deep network features, while losing predictability, are going to actually be really valuable. And finally, just sort of some of the ways that we hope to push this further. You can customize, like the magic in this approach is in the measuring. It is not actually in the dyes themselves. And I've explained experiments that had a few different dye panels in this. And so we're working with researchers to customize the dye panels in different experiments to the biology that they care about. So in this case, this is now out as a preprint. Somebody created a specific dye panel for lipocytes, but then used the same morphological profiling approach, and they're calling it Lipocyte Profiler. We're also hoping now to be able to query experiments against one another. So now that we've done some cell painting experiments where we've looked at drugs and some where we've looked at genes, saying, can we take the features that we measured in one experiment and look at all the compounds and see how the compounds are similar to all the genes? And so where this work is going forward, but we've found at least a few connections where something that is an inhibitor looks anti-correlated with overexpression of the gene that we know we care about. And in general, the can we predict experiments from one another? Greg Way is a postdoc who is now going to be starting his own lab next month. Did an experiment where we had cells that were CRISPR'd for various perturbations, and we split them into 2 plates, and on one plate we did cell painting, and the other plate we did measurements of the cell cycle and of viability with very specific dyes for those particular features. And then Greg trained a network to see could we recapitulate those phenotypes and figure out which CRISPRs caused which things, like cell cycle differences or DNA damage, just from the cell painting. And a number of the features are actually really predictable from cell painting, indicating that we could use cell painting and not have to do these other more specific phenotypic measurements and just sort of infer them from cell painting data. So just to wrap up, hopefully what you're thinking at this point is, this is all super cool. How can I learn how to do this, and where can I go for help? This methods article, and I will make sure that this slide deck gets the organizers to send to you guys, was a joint methods paper written by about 10 labs in the field on how to do the various steps here and what the important steps are in doing one of these assays. And after writing this paper, they came together to form what we call the Cytodata Society. Their annual meeting will be in October this year, which is a society of labs that work together on these sorts of experiments and are very welcoming to people coming in to learn to do it for the first time. And finally, if you have questions about image analysis for profiling or for CellProfiler or for anything, we invite you to come to forum.image.sc. Where 50 different softwares all share one common form for help, and you can upload a picture and say, I need to find out this information. How can I do so? And you have experts from all of those tools who are there together and can help you sort that out. Finally, just want to thank all of the people who did the work here, the members of my lab and of Ann Carpenter's and the Carpenter Sing Lab. And thanks to our biology collaborators, the people who funded it, and you guys for having me. Thank you so much.
Thank you very much, Beth, for the wonderful talk. We will be Shall we take some questions, if any? Let me see if there is any question in the chat window. I don't see them. Okay.
Mhm.
So I can just ask a question if there are no questions. So in any kind of your deep learning or whatever the profiling methods, so you
Mhm.
had mentioned for in one of the point you had mentioned for a one single cell, you can roughly around measure 5,500 features, right? So how do
Mhm.
you basically are you going to consider all of the feature into account to identify the phenotype, or is there a specific way that it would do?
Great question. So we typically we start with 5,500 features, and then we do a few rounds of filtering. So one thing we do is we just throw out any features that don't vary. So if 95% of the things all have the same measurement, that can happen with things like solidity, how many holes are there in the cells, we throw that feature out. We then also, because we don't want, we can have often multiple measurements that give very similar results. You know, the standard deviation of the intensity in the cell can be pretty close to the median average deviation, and the mean is often close to the median. If two measurements are more than 90% correlated, we throw out one of them as well. And so we typically start from 5,500 features, and we end up typically with sort of 300 to 1,000 after that filtering. And at that point, that's what we use to ultimately do our profiling.
Okay. So it's basically you try
All of
to.
that is laid out in the Nature Methods article that I mentioned.
Okay, thank you. So participants, if any of you guys have any questions, I see a question.
Is this pipeline as?
Shall,
Oh, sorry. I'll let you continue
yeah.
that.
Is this pipeline as efficient in other samples like animal tissue samples?
Yeah, so moving the profiling approach into tissue is absolutely something that we've played around with. The biggest challenge just typically is can you get accurate cell boundaries within a tissue? It's much harder. But there's absolutely no reason that the same approach can't work there as well. It's just because it's harder to get accurate boundaries. It's not something people have tried very much. But in theory, it's not particular to fluorescence, not particular to cultured cells. It's just, you know, getting as many measurements as possible is sort of the real magic to the trick. Mm-hmm. I had one slide that I skipped that I'll just go back to since seems like we might have time, which is just to say one thing we are working to do is to now create the world's largest public cell painting data set so that people can do these sorts of querying experiments. Because there are no public cell painting data sets that are queryable in the same way. And if people want to be able to do this on their samples, on the biology they care about, and then match it against a larger database of cell painting data. We're working with actually 10 different pharmaceutical companies, 3 nonprofits, and ourselves to create a data set that has over 100,000 compounds and several tens of thousands of genetic perturbations in order to be able to have that be a publicly available data set that people can use. And so that will be private until next year because the people who helped make it get first access to it, but we're really excited for everyone to be able to access it in the future.
Yeah, this is great. Participants, you guys have any questions for Beck? You can also unmute and ask if you wish.
It's either really clear or really confusing.
No, it was amazing talk actually.
Mm-hmm.
I hope participants enjoyed the session.
Mm-hmm. Well, thank you so much for having me. I really appreciate it. I know it's on the later side there, so probably also folks are just tired, but it's been really great to get to tell you some of these stories.
Sure. Akshaya, should we wait for some more time? Oh, okay. So, Beth, we go for a screenshot,
Mm-hmm.
a picture, a traditional way. So could you please unshare your screen so that we will
Yeah.
have the layout?
Mhm.
So we request all participants to come on the video. Is it done, Ruchika?
Yes, we are done.
Okay. Thank you very much, Beth, for your time.
Right. Thank you so much. And again, I will make sure to send the slide deck along, but thank you so much. Bye, guys.
Thank you so much. Catch you later. Yeah.